back to my house. Thank you. Thank you.
I'm going to say a few more things about meta-analysis, and I'll be covering some of the ground that was mentioned earlier, but it doesn't hurt to hear some of these topics twice. So first of all, scientific studies are often repeated. We know this, and we regard it as a good thing. And why we do this, it's not necessarily insulting some predecessor's work, but rather we work in different systems. We address the same question, but not in exactly the same way. And in ecology, maybe meta-analysis started in the 1980s with Tom Schainer's analysis of field experiments on interspecific competition. When he did a literature survey, he found that there were 164 published experiments. And that was a surprise to everyone, that there were that many. And here's another examples. Recently came across Alison Bell's paper in which she summarized 759 published estimates of the repeatability of behaviors in the animal population. So studies are repeated, often lots of times. Why meta-analysis? Well, a method is needed to summarize results from these multiple studies. There are some sort of tragedies from the past that make it plain that this would be a good thing, or it made it plain long ago that this would be a good thing. So, my parents' generation, they all read Benjamin Spock's book, Baby and Child Care, and he sold 50 million copies. And in it, he has this quote: "I think it's preferable to accustom a baby to sleeping on his stomach from the beginning, if he is willing." And other pediatricians have made similar recommendations. And from the 1950s to the 1990s, one of the alarming things about babies was that a very large number of them died of something called sudden infant death syndrome. And in the early 1990s, researchers realized that the risk of sudden infant death syndrome decreased by 50% when babies were put to sleep on their backs rather than on their stomachs. And so subsequent education campaigns basically stopped this early recommendation. But in sort of doing the analysis, people realized that actually the data was out there much sooner than the summary came forward. The data was already in the literature that would have supported this decades before the news actually came out, and it's thought that if we more routinely synthesize results from the literature quantitatively, then there would be benefits. Now, in the old days, a review article is the way that this was done, and it was usually some expert assembles all the studies that he or she can find, and then reads them carefully, and then writes a review synthesizing the results of those papers and reaching some overall conclusion about what they're saying. And the literature is populated with those kinds of analyses, and very smart people do this reviews, and then the results really are synthetic and can move the field forward. New hypotheses, new paths of research, and so on. Well worth reading those. But there are issues from this methodology, and that is that, well, one is that it's hard to generate from this a quantitative summary of research findings. How big is the effect that's being reviewed? How would you even go about summarizing that? And the other is that there's the potential for bias. So Linus Pauling is the only individual to have been awarded 2 unshared Nobel Prizes during his career. He wrote a book in 1986, How to Live Longer and Feel Better, in which he talked about the benefits of large daily doses of vitamin C to reduce the risk of contracting the common cold. And in this book he cited 30 studies that supported the idea, and no studies were cited that did not support his idea, which seems biased. Most reviews aren't this biased, but how do you prevent it? What's the methodology to minimize bias? By the way, How many of you take vitamin C when you have a cold? Quite a few maybe see. So the idea lingers. There have been meta-analyses evaluating the benefits of vitamin C for contracting cold or for speeding through the process and recovering from a cold. And the basic upshot is it has no effect at all. There are a few special circumstances in which it might have an effect. Tom Shaner's analysis, though, was a kind of analysis methodology that's referred to as vote counting, where basically you just take all the papers that you read and divide them into two piles. Yes, a significant result, no statistically significant result. And then the proportion of studies that voted for the effect, sort of the measure of how prevalent the effect is. So here's the statement of the result. An overwhelming fraction of experimental attempts to detect interspecific competition in the field did so. 148 of 164 studies, or 90%, demonstrate some competition. 110 of the 148 studies record changes in numbers through local births, deaths, or migration. So clearly, interspecific competition is not a peculiar result. But can we say more? How strong would it? For example, vote counting won't give us that result. So by counting only the statistically significant studies, vote counting ignores all the quantitative information in those studies about magnitudes of effect. It's also conservative. Votes are affected by the power of individual studies, which might be weak. And significance level by itself doesn't indicate whether two or more studies obtain the same outcome. The magnitude of the effect is even downplayed by vote counting. The emphasis is on, is there an effect? Yes or no? It's difficult to quantify the effects of publication bias in that vote counting context. When I read this paper as a graduate student, I decided that it's a hell of a lot of work to do an experiment on interspecific competition. So nobody's going to actually carry out a study, an experimental study, unless they have pretty good evidence already that there is competition between these species. So my view was that when there's indirect evidence for interspecific competition between the species, an experiment will usually confirm it. But what that says about competition in general is not clear. The method's unable to weigh the effects, the strength of different studies at different sample sizes and different power. Here's an example of the limitations of vote counting in a case where an effect was small but biologically significant. So the Anti-Platelet Trialists Collaboration conducted a meta-analysis of 142 randomized experiments testing whether taking aspirin or other antiplatelet medication following a stroke or myocardial infarction reduced the risk of future stroke. Total number of individuals participating in all of these experiments combined was like 70,000. The vote. 19 of 142 studies showed a significant effect. That suggests that maybe it's not very important at all. But if you just looked at the numbers all combined, 15% of patients in the control groups had subsequent vascular events, compared with more like 11% in the treated group. This is not a large effect, but aspirin is so cheap and has so little side effects that nonetheless this treatment could save many lives. So meta-analysis, the analysis of analyses. Meta-analysis is the statistical synthesis of results from a series of studies. So a systematic review is the review itself, and the meta-analysis is the statistical component of the review. It involves compiling all known scientific studies estimating an effect and quantitatively combining them to give an overall estimate of that effect. The idea of meta-analysis is that it would allow you to generalize, allow the field to generalize and move forward, and get an idea of just how frequent, how important, how consistent, how large are the effects across a variety of systems. meta-analysis gets past the occasional sensational results, the ones that you read about in the papers, to an overall assessment of what the effect really is. So it's an attempt to be completely objective in the summary of data. The idea, the method, I believe, came from medical research. And an advantage of medical research is that all of the studies are done on one species, humans. And so there's a reasonable amount of consistency among different experiments. Despite this, and despite the fact that not only are they using the same species, but they're using exactly the same medicine as well, the outcome of these trials, the estimated effect size of these trials, varies greatly from study to study. And a lot of that variability is sampling error. And some of it is true heterogeneity in populations and in methodology, but a lot of that is just sampling error. Here's a portion of the results from the platelet trialists collaboration. So it's called a forest plot. It lists each row is a different study or species in our case, and what's shown in the forest plot is a variable that summarizes the association between treatment and response. And this is an odds ratio, so it's the odds of success. No, it's the odds of another event, another myocardial infarction or stroke under the treatment compared with the control. And so less than 1 means that there is a benefit, and greater than 1 means that it's worse to take the aspirin than no. And so a lot of these studies are hovering, so this is the effect size of the individual study, and then the line across is the 95% confidence interval. And you can see across these studies how many of these individual studies do not, the coverage of the confidence interval includes the value 1, which is no difference between treatment and control. So individual studies may not show a statistically significant event. But if you summarize all the studies together, you see that there's definitely a trend across all studies, and that there's a reasonable effect size when one averages across all those studies. Now, most of us don't work on humans. We, instead, probably none of us in this room even works on the same species. And so it's more of a challenge to try to summarize those kinds of results, because they're highly heterogeneous from study system, the study species, but even the population that's investigated might differ from place to place. And we can ask ourselves, what exactly are we estimating when we summarize effect sizes across all studies? So I'll talk a little bit about how that's handled in the statistical methodology, but I thought I'd give a couple of examples. These are pretty early examples of meta-analysis. This one's in psychology. And I want to use it to make just a couple of basic points about meta-analysis. So it's all on humans, but nevertheless there's a fair amount of heterogeneity among the studies. And it was an attempt to synthesize all of the literature on lunacy, which literally means the moon influences human behavior. So many people actually at one time believed, and maybe many still do, and there's all kinds of movies and books that talk about or echoes the legends of strange events in the past. Monsters like werewolves and vampires, and their behavior and their activity is linked to the lunar cycle. Lord Blackstone, an 18th century English jurist, was the first to define a condition of madness exacerbated by the lunar cycle. A lunatic is properly one who hath lucid intervals, sometimes enjoying his senses, and sometimes not. And that frequently depending upon the changes of the moon. So when they carried out their meta-analysis, Rotten and Kelly found that half of university students believed that people act more strangely during a full moon. In 1995, Vance reported that as many as 80% of mental health professionals believe that the full moon can alter individual behavior. So this is not the lunatic fringe. So Rotten and Kelly carried out a meta-analysis of studies that correlated homicidal rates, psychiatric hospital admissions, suicide rates, crisis calls, etc. in association with the full moon. And so that's what I mean by heterogeneous studies. They basically just tried to include any kind of study using any kind of metric that had attempted to equate or associate full moon with human behavior. And the results are summarized in this funnel plot. So sample size on a log scale is shown along the bottom. And then effect size is along the x-axis. And even though not even all these studies involve associations between continuous variables, usually there's a way of converting any kind of an effect size in a study to something like a correlation coefficient. And so the correlation coefficient is often used as a measure of effect size, and everything might be converted to it if there's sufficient information in the original publication. So there's the scatter of points, and there's the zero line, and we don't have to do statistics to know that there's absolutely no association in analysis between effect size and sample size. And there's no effect size average different from zero in this case. Now, one of the reasons why I wanted to show this example is look at all of the studies that are associated with a sample size of 12. Can you think of why?
12 months a year.
12 months a year, yeah, exactly. So that would represent basically 12 lunar cycles, approximately. And what is the duration of the lunar cycle, exactly? 28 days. So what that means is, say, in a particular year, if the full moon is on a Saturday, when's the next full moon going to be? Saturday. And when is the next full moon going to be? On a Saturday. It takes a while to dissociate day of the week with the lunar cycle. These are all studies of 1 year. So the correlation across 12 months, essentially, between human behavior and the lunar cycle. But, there are more hospital admissions, more suicides, and everything happens on weekends. And so the extent to which you've got a positive or negative association is in large part dependent on whether your 12 months of study involved the lunar cycle landing on a weekend or not. And so I show this just to make one point, which is that when you go out and survey the literature, there's a lot of crap out there. And then, careful decisions need to be made about exactly how those data are. Well, first of all, whether they should be included at all, and then secondly, how to incorporate them. I mean, in this case here, the sample size isn't 12, it's 1 for all of these studies. So one has to think about these things when one summarizes from the clinical studies. So how does one go about doing meta-analysis? Well, the really important step is to define the question, because this is going to determine what literature you survey. And the question you ask could be a really narrow question, like, does aspirin reduce incidence of myocardial infarction? Or alternatively, a more heterogeneous set of studies. How about a heterogeneous set of variables? So here's a much more general, how much genetic variation exists in populations for behavioral traits? That would be a much broader study. And then you have to decide, well, what quality of studies will you include? Only experiments with controls? Well, if it's an experiment, it's going to have some kind of control. But how about experiments that use randomization only? Experiments that are properly replicated? Experiments that use blinding? there's two ways of approaching these kinds of questions, and one is to keep the scope relatively wide, and then later on ask, is there any difference in the average effect size between studies that incorporated blinding and experimental studies that did not? So that's something that meta-analysis allows you to investigate, actually. Then there's the literature search. Now, the question came up, how many of you have tried to find unpublished data before? It's not very easy. But if you want this meta-analysis to succeed, you're going to have to make the search exhaustive. Easily found studies are different from those you cannot find easily. And in the workshop on Thursday, we're going to analyze a data set that was compiled by a group of people on a behavioral trait in birds in relation to dominance and plumage. And the study's kind of interesting because last year I found this paper like just before the workshop and I incorporated it. And the reason I did that is because previously I had the workshop on their previous publication. And in the interim between the first and their second publication, both meta-analyses, they had found many more unpublished studies. And so you'll be able to look at the consequences and whether there's a difference between those studies that were published and those that were unpublished. How did they find the unpublished studies? They just wrote everybody. They just wrote letters to everybody. So, as we saw, there are some methods to account partially for publication bias, and I'll reiterate some of those, but they don't replace a truly exhaustive survey. And then I already mentioned the question of whether you hold your nose and include everything, including studies of poor quality, or whether you have well-defined criteria for inclusion of studies. And well-defined criteria to if you use criteria that are not completely objective and well-defined, then there's the potential that your meta-analysis will incorporate bias that you might not intend. All the data obtained should be independent, but non-independence of various sorts always creeps in. And you'll find that in the meta-analysis on Thursday that there are multiple data sets from single authors. Is that one study? Is that 5 studies? They're done in different years. How do you incorporate all of that heterogeneity? A single study might provide measurements on multiple species or multiple responses on the same species. Should you include all of them? Or should you take a single summary measure for a particular study? You'll typically find that one or a small number of species or systems will be overrepresented in the literature. Should you treat them all as though they were some random sample from nature, or what? Well, there are pros and cons of either approach. And here's another one that again addresses the question of how heterogeneous your study should be, how specific the question should be. Are testosterone levels and aggression correlated in human males? And this analysis combines many different kinds of data sets. And some of them are quite amusing to read. So, levels of testosterone in prisoners convicted of violent crimes compared to those convicted of property crime. Levels of testosterone in university students compared with their answers to questionnaires that asked them for levels of agreement to statements like, if somebody hits me, I hit back. Levels of aggression in Kung San males as determined by counting their scars and sometimes still open wounds in the head region. Drunken Finnish spouse abusers compared to drunken Finns drinking quietly in a bar. Members of rambunctious fraternities compared to responsible fraternities. So these are all published studies. And in this meta-analysis they just said, we've got to include it all. Otherwise, how do you decide yes or no, this study, none of them are experiments. They're all observational studies. How do you say, we won't include this one, and that's not politically correct. We'll include that one. So there's the funnel plot for their study. Again, they reduced everything to a common measure of effect size, the correlation between treatment and response, testosterone and response. And I have huge misgivings about using the correlation coefficient as an effect size, because the strength of the correlation always depends on the range of variation you have in the explanatory variable. You know, the range of variation in testosterone included in every individual study varies, and you know that's going to affect the strength of the correlation, even if the response variable is identical in every case. Welcome to meta-analysis. Anyway, there is a positive effect size overall in this case. Looking at these scatter of points, so the way a funnel plot works is, here's the sample size, there's the effect size. And what these upper and lower curves mean is that they represent the critical values associated with rejecting or failing to reject the null hypothesis of no effect. So if you're outside that line, it means the result was statistically significant. If you're inside the line, it means the result was not statistically significant. And you can see there are a number of statistically significant results. It's called a funnel plot because as your sample size goes down, the variability goes up. The other thing that I think is important to remember, and I guess it's really driven home by meta-analyses, is that look at the heterogeneity of responses. I mean, everybody's trying to measure the same thing, perhaps using a different methodology and a different population, but the same species. And look at the heterogeneity of results. And you know, sometimes there are statistically significant results on the wrong side. That's potentially sampling error. So it's one reason why it really is important to synthesize results from the literature. You know, this is probably the one that made the papers. Less testosterone makes you more aggressive. So it's always fun to read the headlines in the newspaper about the results of studies, but it doesn't mean that that's the trend. So one of the decisions in a meta-analysis is now to calculate an effect size that can be combined across studies. These are the correlation coefficients. Can be used, and sometimes it's the only thing that can be used because of the heterogeneity of methods that's been used. In a highly homogeneous set of studies, like clinical trials testing the effect of aspirin on myocardial infarction in humans, you can use the odds ratio on every single one. Another one is the response ratio, the average effect in the experimentals versus the control, which eliminates the units. And then a Cohen's D or Hedges G, that's the conventional method for measuring the magnitude of the difference standardized by the amount of variation that's present within groups. So it's essentially a measure of how many standard deviations apart the two groups are. And that again allows you to use studies that have response variables that are measured in completely different units. More or less the same thing. It just corrects for a small sample bias.
You don't.
Yeah.
In that funnel plot, or did they say? Would you say that there's a propagation bias there because you have you don't have a lot of studies with it's not really very symmetrical.
Oh, you're absolutely right.
And did they address that?
So, if the mean effect size is something like this, maybe 0.2 perhaps, what you can see is that there's most of the observations, the frequency distribution of results should be highest at 0.2 and reduced uniformly to either side. But that's not really what we see. At small sample size, we don't see any results in that range. So absolutely, there's propagation bias. And one of the ways that might be addressed is to plot the relationship between the mean effect size and sample size. And generally, the most believable results are the ones that persist when sample sizes get very large. And so in this case, that might be approximately 0.2, whereas you might get 0.3 if you just averaged all the studies without taking propagation bias into account. Here's a third example of a meta-analysis. Alpert used the log response ratio to compare marine reserves to comparison areas. or the same area before the establishment of the reserve, and the abundance and diversity of fish and or invertebrates. So biomass, diversity, density, body size of the organisms. So sample size in each case is along the x-axis. Effect size, this log of the response ratio. No effect is 0. And these are the kinds of results that persuaded people that marine protected areas actually do what they're supposed to do. They work. Whereas it would be hard to draw that conclusion from just a single study or a very small number of studies. So meta-analysis is a statistical analysis, and we're going to work our way through the stats almost by hand, although not quite in the workshop this week. Although we will use also, if you get through it, using our package called Metaphor, which is truly amazing. And it's free. I can't believe the amount of time people devote to creating these packages that then just give you. So there's two sort of classes of meta-analysis. One is called the fixed effects, and the other is called the random effects. So fixed effects is commonly used in meta-analysis. In medical studies, you'll never see fixed effects analysis in ecology or evolution, most likely. And that's because a fixed effects model assumes that all of the multiple studies are actually estimating the very same mean. That the mean is identical in every population in which this investigation is being carried out. Well, we all work in different systems or different species, so that assumption is just not on. But in human studies, that's probably not on exactly in human studies, but it's certainly perhaps the only case in which it would be justified. It assumes absolutely no heterogeneity among studies. Unlikely even in human trials, but certainly not in our field. In the random effect size, the assumption is that there's random variation present among means of studies in addition to sampling error. So this is similar to the random effects model that we already analyzed in the linear models, the mixed models workshop. So the idea is that individual studies are all estimating different treatment effects because every study is assumed to be different from every other study. And in this case, what does it mean to take an average of all of those studies if none of the means are the same? And the answer is, well, the focus is on the central value, the typical value, or the mean of means. The idea of a random effect study is also to understand that the distribution of effect sizes across systems. How variable is the effect size in different systems? So this is, again, very similar to the fixed effects linear model and the mixed effects linear model. In the fixed effects, you're estimating mean plus a random error, and this is the true effect size, and it's the same effect size in all of the studies. Whereas in a random effects model, there's the grand mean, but then there's the individual mean of each particular study system, I, and then the random error. And the computation is different in the two cases because each study is weighted in a different way depending on how far away they are from the true mean and their individual sample sizes. Unfortunately, LMER and LME, some people, I've seen analysis where they actually do this, but apparently LMER doesn't work for meta-analysis because it gets the weights wrong. And so you should actually use a dedicated meta-analysis package like Metaphor, which models the heterogeneity. Once you have your meta-analysis, you can start to look for effects of study quality. Our effect size is different on average between studies that include blinding and those that did not. So people have done such analyses in human clinical trials and shown that generally effect sizes are smaller when blinding is incorporated than when it's not. But again, this part of even if you're summarizing experimental studies, this is an observational study. The meta-analysis is an observational study even when it's summarizing experiments. And so there's the potential for confounding variables. Usually studies that don't include blinding have other problems as well. One can also look for interesting associations. For example, if you include a whole pile of studies in your analysis, you can ask, is the effect size different in men and women? So you assume that there might be some heterogeneity, and then you can possibly investigate it. In the history of meta-analyses in ecology and evolution, Gurevich followed on Tom Shainer's meta-analysis of inter- and intra-specific competition, but they this time went through it and did it with a quantitative analysis. And Tom Shainer was hurt by this, that it was actually necessary for someone to go and redo his vote counting method, but well, the methods had changed for meta-analysis, and they wanted to explore more deeply what effect sizes were. And so they were able to quantify the mean effect sizes across these experiments, and they were able to look at the differences between the outcomes of the experiments that had what they called serious flaws versus those that had reasonably good designs. And I can't remember exactly how they categorized individual observations. I mean, it looks like there's more variability in the answers when the studies are flawed, but this is a confidence interval, and I don't know what the sample sizes are that are contributing to it too, because that would also affect the confidence interval. They showed in this study that generally if you cage your animals, you get a much stronger effect than if your animals are open and can move freely in and out of the study plot. A couple of other points or issues in meta-analysis. One's the file drawer problem. That's basically just a name for the problem that in file drawers, okay, do you know what a file drawer is? It's one of those things that has these drawers and there's paper inside. So people used to have them. Now it's the file folder problem. But the idea is that sitting on computers somewhere are the unpublished studies that you want and aren't published. And as we've seen, funnel plots can give an indication maybe as to whether bias is present or not. Funnel plot are the effect sizes for the relationship between song complexity and reproductive success. And in this study, they included published studies, those are in black, and they tend to show a more positive effect size than unpublished studies. Unfortunately, the symbols aren't coming clear, but these are studies that are just sort of thrown on there by this trim fill algorithm, which tries to fill in the asymmetry and the shape of the effect sizes around the central mean. So there are imperfect methods that might help. Another way of trying to see whether publication bias might be a serious problem is the fail-safe number, and it's how many missing studies would be needed to change the result. So in this analysis here was a large meta-analysis of the effects of alien plant species on native communities, native plants and native animals. And they included in their analysis the number of studies that would have to be sitting in file drawers, unpublished studies, to change the most significant results of this meta-analysis. And they concluded that there would have to be nearly 40,000 unpublished studies to change their result. And the idea is that you look at that number and you go, nah, that can't be. And so you conclude that the effect is real. But you guys are going to be publishing soon, if not already. And when you carry out a meta-analysis, if you ever carry out a meta-analysis, you'll be frustrated by some papers that don't actually include enough information for you to calculate the effect size that you're going to use across all the studies. So don't be in that group. If you publish your papers, make sure there's enough measurements of means and standard deviations and standard errors. So always give a size of effects and standard errors. The p-value by itself is useless. In meta-analysis, in fact, a p-value is useless. Meta-analyses don't actually use the p-values that you calculated in your publications and that you proudly show in your ANOVA tables. And I've always found that this is, in my mind, a measure of just how useless p-values really are. That mostly what meta-analysis is attempting to do is to find out how big effects are, not whether results are statistically significant or not. That's vote counting. In general, meta-analyses will only use the effect sizes that are in your published studies, and the p-values, who cares? So give the estimates of your coefficients of means and standard deviations. Always indicate your sample sizes and or degrees of freedom. Better yet, publish the data. Put them on dry ash. Consider a meta-analysis for your first chapter. So, the first chapter of a thesis is often a review of the literature in your chosen area. Well, if you're going to review the literature on a particular topic, keep a pencil and a notepad next to your computer and write down some of the numbers that are presented in the papers that you're reading, so that you can compare them. You can actually come up with a quantitative summary of the literature so far for the question that you are interested in. I mean, even being able to show how uncertain the effect size is from the existing literature is something that would justify the work you're about to perform if you're going to be doing a similar kind of study. There's now lots of information out there on how best to carry out a meta-analysis, and if you're going to do a meta-analysis, you should definitely explore this literature. So the Prisma statement was already mentioned in the presentation earlier. And there's sort of a standard methodology, not just for the meta-analysis itself, but for the entire systematic review, of which the meta-analysis is the quantitative part. And there are these flowcharts for each of the steps of the systematic review and then the meta-analysis. And the idea is that you would publish all of these decisions with your meta-analysis so that you could explain to everyone exactly what decisions you made at each stage. And that is a key component of the best practices for systematic reviews and meta-analyses. So the Prisma statement provides a detailed checklist of things for you to go through. So I would certainly start there if the idea ever occurs to you to carry out a review. Meta-analyses tend to be cited a lot too, so it won't hurt. There's a package in R called Metagear, which is meant to facilitate the compilation of data from the literature to be used in meta-analyses. I've never used this. If any of you explore this, let me know whether you found it useful. Traditionally, meta-analysis is the analyses of effect sizes and their standard errors, their uncertainties, usually determined by sample size, and then quantitatively comparing them across all studies. But the world is changing, and more data sets are now accumulating at repositories like Dryad. It's now possible to do a meta-analysis of open data sets. So don't just use the values that were calculated by the paper, go right to the data and start again. So this was the first paper that I encountered which attempted to identify some of the issues and to construct a kind of framework for open data analysis. meta-analysis. And I wanted to show one other thing, if I can find it. So registering your study is something now that happens routinely in medicine. So if you want to carry out a study and you want to be funded by NIH, then you have to register your study so that there's a record of what you planned to do, and anyone conducting a future meta-analysis would at least be able to go, these guys carried out a study. It was funded by NIH, but they didn't publish anything. I'm just going to phone them every week until they give me their data. So registering has that advantage. The other advantage of registering is it forces you to state your hypotheses up front. And therefore, it's possible to go to the publication and say, well, what were the results of those specific hypotheses? And then what came after? New ideas, exploratory analyses, and so on. So this is already happening for human trials, but this was a paper just out this year in PLOS Biology that proposed an animal study registry. So come on, you plant people. We don't have one yet for you. So that's the introduction to meta-analysis. Next week, multivariate analysis. Something I know you're all curious about. So there's a handout on the website. So go read the paper. It's kind of a complicated paper. It's a aquatic ecology paper from John Shuren and others, and it uses a series of multivariate analyses. And I guess the main point of my asking you to read it is so that you can evaluate, what did we learn? Did this give us new insights? Is multivariate analysis useful? Did you understand it? We already have our presenters and our moderators. Fantastic. I will, at the workshop on Thursday, I'll take you aside and give you the usual rundown on expectations for the presentation and moderating.